Okay, good morning. We have a new session today, and we have with us Enrique Marcos, sorry. He is a researcher at the Institute of Molecular Biology of Barcelona, the IBMB. He's actually the head of the Protein Design and Modeling Group. He is a chemist. He did a PhD in molecular modeling of enzymes in the Institute for Advanced Chemistry of Catalonia at the Spanish National Center Research Council. Later, he did a postdoc at the University of Washington, where he worked on the Novo design of protein folds. A few years ago, he came back to Barcelona, first in the IRB, the Institute for Research in Biomedicine, and later at the position he occupies now at the IBMB as a Ramon y Cajal researcher. So thank you very much. The talk today from Enrique is going to be protein structure from prediction to de novo design. Thank you.
Welcome. Okay, thanks Arnau for the kind introduction and for the invitation to come here to talk to you about what we are doing. It's a real pleasure to talk to new students that embark on this new field. That it's a very exciting field, and as you will see, it's a very exciting moment to actually to start to work on this. So today I'm Enrique Marcos, and then we're working now at the IBMB, where I started my lab over 3 years ago with a Ramon y Cajal position. And then I started a new research line where we try to combine computational protein design with experimental characterization of the designed protein. So we try to combine the computational experimental approaches to design entirely new proteins from scratch for achieving novel functions not seen in nature, and then always in close connection with experiment. So let's see. Okay, so today I have divided the talk in 3 parts. So in the first one, we'll talk about key concepts that most of you probably already have about protein structure and previous and current approaches for predicting structures from sequences. And then I will show you how we can use these methods to actually design new proteins, what is, I would say, the opposite problem to prediction, so design entirely new proteins. And then I will show you a little bit about one of our research lines that we have in the lab, where we combine structure prediction with protein design to actually develop a new concept of antibody proteins and antibodies for different applications. Okay, so as you will know, proteins are the molecular machines that sustain life, right? In all the kingdoms of life, they have a huge variety of structures, and these allow them to actually perform a huge variety of functions. So they allow us to digest food through enzymatic reactions. They allow to transport molecules across the body, for example, transport oxygen. They allow to transport molecules across the cellular membrane. They can harvest light to produce energy in plants. They provide our protection against pathogens through antibodies. This is in our immune system. So this is the molecular machinery of life, and they can perform this huge variety of functions thanks to the huge variety of sequences and structures that they can adopt. So just the key concept of a protein structure. So if function is encoded in the structure and the structure is encoded in the primary sequence, that is the sequence of amino acids. So we have 20 building blocks with different physicochemical properties, which allow to control the overall structure of the protein. So as I said, so in proteins we have a polypeptide chain formed by a given sequence of amino acids with different properties. These sequences fall in what is known as secondary structure elements. So some stretches of sequences have more propensity to form alpha helices, others beta sheets. Then these secondary structure elements are arranged in the 3-dimensional structure to form protein domains in with different shapes. And then for many proteins, protein domains have to arrange in different ways to actually build a quaternary structure, what allows to generate much more complex functions. And then for many years, researchers have been intrigued about how to determine the three-dimensional structure of proteins. So essentially, we have three main experimental methods to determine the structure. We have X-ray crystallography. This was the first one. Then we have nuclear magnetic resonance and cryo-EM. We have, in the last few years, there has been a revolution in the cryo-EM field, which allows to actually determine much more complex structures, larger assemblies, and even incorporate protein dynamics. So the first structure of a protein was determined in 1959. This is the first crystal structure, and it was a huge advance in the field. And then since then, so all these experimental methods have been improving over the years. It has become much more accessible to many different labs in the world. We have advances in computing power that allow to process much more data to actually solve these structures. And then over the years, so there has been an exponential growth in the number of structures that have been experimentally determined. And all these structures are collected in what is known as the Protein Data Bank. This is an open access database that allows any researcher to access to any protein structure that has been ever solved. However, determining experimental structures, despite all this progress, is extremely hard, and you need many resources to actually determine the structures. And it turns out that it's much simpler, much easier to actually identify new protein sequences for any given organism. And there has been millions of sequences across all the kingdom of life. And there has been an historical gap between the number of structures that have been determined and the number of sequences that have been identified. And nowadays, we have a gap of about 1,000-fold. So this means that for a huge number of sequences, we don't know the structure. And this is a very important limitation, because if we want to understand protein function for applications in disease, for biotechnological applications, we need to know the structure. So, so there has been the critical challenge in the field, I would say, for the last 50 years. So to bridge this gap, there has been a strong motivation to actually develop computational methods that allow to predict three-dimensional structures just from the primary sequence. This is what is known like the long-standing protein folding problem. And this is a very hard problem. Because essentially what we want to do is to actually, for a given amino acid sequence, try to identify what is the lowest energy minima. So there is the Anfinsen hypothesis that the native state of a protein is in the lowest energy minima in the protein folding energy landscape of all the possible conformations. But the possible conformations of a protein is an astronomically large number. So there has been different ways to actually tackle this. But the key principle of all these methods is that what they have been developing is algorithms to actually sample protein conformations, what is known as the conformation, to perform conformational sampling. And then it has been a strong effort to actually develop what is known as energy functions or force fields. Okay? So these are mathematical equations that describes the physical chemistry of the system. And then for a given protein conformation, it describes the overall energy of the system, makes an approximation based on the different components of the energy. So for example, electrostatic interactions between charged side chains, van der Waals interactions between carbon atoms, for example, the energy of disulfide bridges. It is a component of the structure that allows to stabilize proteins, and depending on its geometry, you can have much more or less stability. There are also torsional preferences in the polypeptide chain, in the backbone of the chain, based on what is known as the Ramachandran probabilities of all the amino acids to adopt different types of conformations in the polypeptide chain. We have hydrogen bonds. So we have many different components that all contribute to the overall energy of a structure. So in general, the underlying philosophy of all the methods is to develop smart algorithms that allow to sample conformations, generic conformations. You make moves into these conformations. You score them based on this energy function. And then if you get improved structures based on lower energies, you can iteratively optimize these structures towards lower energies. So, so in the last 30 years, I would say, there has been a strong development in the field to actually develop improve computational methods for conformational sampling and computing energies for scoring. And all these methods are grouped into two main families. So one is, and this is the, would say the easiest one, this is well, template-based structure prediction, or what is known as homology modeling. So the key idea is, thanks to the sequencing of many proteins, right? So you can have many homologous sequences that you may have from your target sequence. And then if you find homologous sequences, or sequences that have enough sequence identity towards with respect to your target sequence, so then you can use these templates to actually bias the solution, right? And then to identify these aligned regions that allow to extract component pieces of the structure of your template, and then bridge. Well, and there are methods actually to bridge these gaps to actually generate a model of the overall structure. So historically, this has been the most successful approach, especially if you identify templates with high sequence identity. This has been a quite reliable approach over the years. But the problem is that for many sequences, it can be hard to identify a template that is good enough, that is close enough in sequence. So that's why there has been also important developments on predicting structures without templates, what is known as ab initio structure prediction. And here the idea is, so you have your target sequence, and then based on this sequence, you can already predict the secondary structure of this sequence. So there are some sequence strings that have some propensity for specific secondary structures. So for example, pieces of these sequences can encode for alpha helices, for beta sheets. So in this way, you can predict residue by residue the secondary structure of the protein. And then based on this information, and thanks to the growing protein database, is it possible to collect fragments from these structures? So this means peptides that can range between 3 or 9 amino acids that are very similar in sequence and local secondary structure. And then with all these fragments, you can assign fragments to each position. And then with all this collection of fragments, then there are simulations to actually to model the overall folding of a structure. Iteratively, you perform insertion of these fragments in a random fashion, and then iteratively, as I mentioned before, iteratively, and thanks to the score function, you can guide the overall folding process to actually go to a model that has very low energy, and it can be a very good candidate as a potential prediction of your structure. But then, so the number of conformations that you can generate with these approaches is huge. And then one of the things that have been developed actually to bias and to guide all this process is also to use what is known as coevolution data. Again, thanks to the huge number of sequences that are now available. So based on what is known as multiple sequence alignments, so identifying sequences that are very similar to yours. So then if you identify pairs of positions that across this multiple sequence alignment tend to mutate at the same time, then you can infer protein contacts in the 3-dimensional structure. Because if two amino acids that are close in the 3-dimensional structure across evolution, they have been changing simultaneously all along the evolutionary history of these proteins, it's very likely that these amino acids are in contact in the three-dimensional structure. So in this way, you can predict contacts and then use these as constraints to actually guide the sampling of conformations. And then, yeah, for all these conformations and with the energy functions, and there are different ways to do that, but these models tend to be refined to actually model the overall details of the side chain interactions of all the amino acids, taking into account all the physical chemistry of the system, and then in this way you can score accurately these models and then collect the lowest energy models as potential candidates for, as the solution of the structure. So this has been the two main families of methods that have been developed over the last 30 years. And indeed, there was a competition, a worldwide competition that is known the CASP, for critical assessment for secondary for structure prediction. And the idea of this competition was to motivate research groups across the globe to actually develop methods like this, and then compete to see which methods are the best ones for predicting a set of structures that haven't been published but are known, right? So it's a blind test to actually assess the accuracy of the latest methods of the labs and see how they perform on presently determined structures. And this was a strong motivation for the field, and in this way, so it was clear, but it was a good way also to benchmark methodologies actually to the same against the same data, which would be the best ones that could be probably adopted for a broader, for broader, let's say, labs. So, but then, so this is the CASP. So now there have been now the last one was the CASP 15, so there have been 15 editions every 2 years. But in 2020, in CASP 14, there was a complete revolution in the field. So if you can see, so this was like a brief summary of what was the result of the previous CASP competitions. So there was, for many years, there has been, so the field was quite stacked. So it was, so the progress was already quite slow. It was very hard to make better predictions with all these approaches I was mentioning. But then it came up a new method that probably you have heard about, that is based on artificial intelligence with all these deep learning methods that can make much more accurate predictions. So this is what is known, well, is AlphaFold. We had the first version in 2018, and now we have AlphaFold 2 that actually performs what is said that is to be equivalent to the accuracy of an experimental structure. So it's extremely accurate in the way you can predict now proteins, and also proteins that can be quite large, I would say. So all these methods, previous methods, tend to perform quite well for the small proteins, but by the moment that you move towards larger proteins or assemblies, the predictions were quite difficult to achieve. But with these methods tend to be quite general. So essentially, the predictions that you can do with these methods can be even below the angstrom. If you overlay the backbone structure of the prediction with respect to experimental structure, you could get sub-angstrom accuracy, what is extraordinary. Even based on the arrangement of the accurate arrangement of the backbone, you can also infer side chain positions that can allow to infer function of proteins. So before, to actually predict functions for new proteins that you didn't know what they were doing, so it was very hard to infer function. But if you have a very accurate structural prediction of all these sequences, then it can be more now easier to actually infer function for novel sequences. So, and the two inputs of the AlphaFold prediction are multiple sequence alignments as before. So, as again, coevolution data allows you to actually infer very accurately contacts in the three-dimensional structure, and also it uses concepts from before to identify templates that have high sequence homology in some regions, and this allows also to guide the prediction process. So in this architecture, it allows to infer all these three-dimensional contacts based on all this information, and then it performs what is known as an end-to-end prediction. So it directly predicts the overall structure. And what is very interesting is that it's also able to predict what regions of the protein are predicted with more or less confidence. So, so for regions that are, this is one is known as the PLDDT. This is a confidence metric that is per amino acid position that allows you to determine which are the areas that you can trust more, and others that probably they tend to be those that are more disordered or more flexible are predicted with lower confidence. And this is very important because then whenever you have a prediction from AlphaFold, you can decide if you trust or not the model, or what regions are more informative than others. So it's important to also to assess also the reliability of the models always. And now one key feature of this prediction, this method, is not only very accurate, it's computationally very efficient. So before all these methods, you would have to perform simulations for days in hundreds of CPUs, right? So you would need access to supercomputers, so it was not broadly accessible, and also some expertise actually to actually perform that. But now you can perform these predictions, I would say, in a regular computer just in few hours, and depending on the memory and the CPU you have, or even if you have GPUs. But in just in few hours, you can get predictions of very high accuracy. And that's why it has been developed what is known as the AlphaFold protein structure database. That the idea of this is to actually, what I was mentioning before, to bridge this gap between identify sequences and solve the structures. So what they did in this database is to actually perform AlphaFold predictions for all these 200 million of sequences, non-redundant sequences that have been ever identified. So now if you go to UniProt, that is the portal where you can find any protein sequence, now you can have an AlphaFold model. You can readily download. So this is very powerful because then you can, if you have already a structural model of any sequence, then you can start to try to infer function or well, it's key information to try to understand proteins. So, but as I said, so you have these confidence metrics that allow you to rigorously assess the reliability of the model. So AlphaFold is accurate, it's computationally efficient, and it's very accessible, as I said. So you can anyone can generate these predictions. And now there are servers in Google Colab that even you don't need to download it, so you can do it online, and you can perform the prediction of any sequence. So it's now available for anyone. Okay, so now that's. So we have this, this basic idea of what is the protein structure and how the field has been evolving over the years in terms of computational methods for prediction. Now I can tell you a bit how we can tackle the opposite problem, what is actually design new proteins. So as I said, structure prediction and protein design are two opposite problems. So essentially, we were saying, so in prediction we have an amino acid sequence, and then we have all these methods, energy functions, we have conformational sampling techniques, we have deep learning techniques to predict structures. So, but the idea is to generate models that correspond to the lowest energy minima of the protein folding energy landscape. But then what we do in protein design is to actually, we predefine protein structures. So we have a target structure that we want to build, and then we have algorithms that allow us to calculate amino acid sequences that preferentially fold into that target structure. So whenever we do, for example, a protein structure prediction of all these design sequences, what we want is the following. So we want to ensure that the sequence corresponds to so that the design structure corresponds to the lowest energy minima of that sequence. And then, for example, we have here the RMSD. The RMSD is the root mean square deviation. This is a measure to assess the similarity between a pair of structures. So the idea here is that when you perform predictions, so each dot, for example, can be a protein folding trajectory. So the idea is to predict structures that the closer they are to the target structure, the lower is the energy. So this would be a good test for assessing the quality of the design sequence for that particular structure. This is what is known as the funnel-shaped energy landscape. So energy landscapes that then tend to progressively stabilize intermediates that allow to guide the folding process towards the target structure. So many methods that were developed for prediction in terms of energy functions and conformational sampling have been harnessed to actually design sequences that fold into target structures. So, but why do we want to design entirely new proteins, right? We want to, so essentially because there are plenty of opportunities, right? So the protein sequence space, if you can think of, is huge. Imagine you have a protein that has 100 amino acids. This would be like an average size of a protein. And then you have 20 amino acids for each position. So it means that you have 20 to the number of amino acid positions. So this is an extraordinarily high number of possible sequences. And if you think about the number of sequences that have ever been sequenced that are available in UniProt, and also the number of sequences that can be reasonably predicted that have been sampled across evolution, all these numbers are tiny compared to the actual theoretical space of possible protein sequences. So this means there is a huge sequence space, a huge number of proteins that can be discovered, can be designed, and may have potential interesting applications. And then what we have been discovered in the last 10 years, actually we can design protein structures in quite many different structures, many different folds not seen in nature, and they can be very highly stable with tunable geometries, are very soluble, they can be easy to produce, so they can have so many different advantages over natural proteins whenever they are re-engineered, for example, for biotechnology or for medicine. This has been more traditional protein engineering approaches. They tend to redesign existing proteins. But the idea is with these Novo proteins, you can invent entirely new ones, and you can identify very interesting properties. So, so basically, just the key idea is whenever you want to design a protein, imagine you want to design this particular protein fold made of a set of secondary structural elements. You define these, and then you have methods actually to generate backbone structures that fulfill all these secondary structure information. You can generate families of polypeptide chains which have this conformation. Then there are algorithms to actually compute amino acid sequences, so identify side chains that stabilize the contacts of this structure. And then, as before, if we have a sequence and a target structure, then we can perform all these, as I mentioned, all these simulations to actually make predictions. And then if you identify sequences that preferentially stabilize your target structure, it's a good compatibility test that then that at least indicates that this sequence is promising for validation. So then often when we identify sequences that fulfill these criteria, then we go to the wet lab and actually experimentally produce these proteins, right? So we can obtain synthetic genes that encode for the design sequence and then express them in bacteria overexpress the protein, purify, and then we can perform biochemistry and biophysical studies of all to assess the properties of this new protein. So with all these approaches, so in the last decade there has been a huge number of proteins, different folds that have been designed of different complexities. Also, even larger assemblies from oligomers to cages for many different applications. So the field actually exploded. And now I can show you, for example, applications that are now being developed for therapeutics, for example. Now that is possible, you can generate now libraries of proteins that don't exist in nature. So libraries of proteins that are predicted to be stable, and then there are high throughput screenings that allow you to assess the stability of a library of proteins. So now, for example, you can see, for example, that you can see in black, this would be the number of structures you have in the PDB for that particular size. And these were design sequences with similar stabilities to the structures in the PDB. And then you can see that you can exceed by hundreds the number of protein scaffolds that you can have now available to actually design new functions. So it really opens many opportunities. So for example, if this approach these libraries have been used, for example, to design inhibitors for different targets. For example, for COVID-19, in the context of the pandemic, there were developed de novo design inhibitors to actually target the spike protein of the coronavirus. And then you have these families of designs, and then with different high throughput screening experimental strategies, you can identify the top binders. Also, for example, they can use, for example, to mimic functions of existing proteins. For example, this is a study in which they were designing mimetics of interleukin-2. Interleukin-2 is a protein of the immune system that has been used also for cancer immunotherapy. But the problem is that the natural protein is very hard to produce and it's toxic when at high doses. So the idea was to generate a mimetic of this protein that wouldn't have toxicity and would be, for example, easier to produce. And this was the basis of a new biotech company that is moving this towards clinical trials. Also, for example, again for COVID-19, these protein assemblies that can be designed for nanocages, they can be used as scaffolds, for example, for vaccine design. So if you have these spherical particles, you can present antigens, for example, to actually develop an immune response and develop antibodies against that antigen. And this was used for SARS-CoV-2, and there was already a COVID vaccine that has been already approved for human use. So it's really now a reality that all these entirely new proteins can be used for different applications. But now there has been, as I said before, so we have this revolution based on AI, and this has been changing again the game of protein design. Before, it was more necessary input from the designer because you needed like very detailed knowledge about the structural features of the proteins that you wanted to build. But now with deep learning, you can automatize many aspects of the process. So now there are called, for example, what is known as hallucination methods that you could, for example, generate new structures from random sequences. This means that if you have, as we said before, for a given sequence, you can predict the structure. But then if you start from a random sequence, that prediction is not going to yield like a very well-folded and globular protein because it's a random sequence. But then you can iteratively optimize this sequence to progressively optimize the foldedness of the protein. So in a way that you perform predictions and the contacts you can predict across the simulation are better defined across the simulation. So you optimize the sequence towards better folded and more globular, more regular structures, and this is already guided by for the prediction process. So in this way, without pre-specification of actually what is the protein that we want to build, you can generate new ones from random sequences. So this means that you don't need you need minimal input information to actually build the structures. You can do even more complex things. That if, for example, imagine you want to you have a functional motif at a protein-protein interface, and you want to build a protein around that functional motif to actually stabilize that. So before you would have to predefine what is the fold that you want to build to actually stabilize that motif, what is a very hard problem. But now with these deep learning methods, you have this motif, and you don't have to think about what is the scaffold that is going to actually stabilize that motif. So the overall process is going to do it for you. So, so essentially, yeah, you can get more robust predictions with, as I said, with minimal input. So yeah, so just for the third part of the talk, after having given you a background of what has been done on prediction, what is being done on protein design, I can tell you a bit about what of the things that we are doing in the lab in the context of all this in this field. And it's about the design of antibody-like proteins. So the idea is that, well, as you probably all know, well, monoclonal antibodies are the largest growing class of pharmaceutical drug. It's being, well, they are being applied for many diseases, including cancer, for immune diseases, even as antivirals. We have seen again for the COVID-19, so many antibodies have been developed. So, and antibodies, well, have this type. This is a scheme of antibody. We have the variable region of the antibodies that is responsible to actually to detect the antigen. So it's not variable because in principle it can be evolved to detect any particular antigen. There are these flexible regions in the variable region, what is known as the CDRs, the complementarity determining regions. These are flexible loops that can be optimized by often, well, mostly by directed evolution strategies to actually be optimized for particular targets. And this has been used for therapeutics, has been used for diagnostics, for example. If you want to detect a particular protein, particular antigen, or it's been used for diagnostics as well as for research also, right? If you want for cellular studies, for example, to detect specific proteins. So antibodies are used everywhere. But the problem is that they have important limitations in terms of production, stability, size. They have several limitations, and for many years there have been people interested in designing derivatives of antibodies that may have improved properties. For example, what is known as nanobodies. These are single domain variable domains, single chain variable fragments. So there have been, since the late 90s, they have been developing many alternative formats for the variable region. But one of these limitations is that, again, they always rely on the naturally existing structure of the antibodies, what is very well conserved. But then, thanks to the advances that we have been doing in the Novo design, so we were wondering why, instead of trying to repurpose the immunoglobulin framework that is known in antibodies, let's try to build new ones. So try to do like antibody-like proteins entirely from scratch. So in a way that we will be able to avoid all these limitations that we always have with natural antibodies. For example, we can generate antibody single domains with tunable geometry, tunable size. They could be designed to be hyperstable, so that you can leave them at room temperature, for example, and avoid a cold chain infrastructure. You could incorporate even different functionalities to what you see in natural antibodies. They could be far cheaper to produce, for example, using bacteria, for example, as an expression organism. You could, for example, avoid relying on disulfide bonds. What often inside the cell, it can be limiting to have dependency of the stability on disulfide bonds because it's a reducing environment. So here the message is that instead of inheriting properties that we see in natural antibodies, we can try to develop new ones with improved properties but inspired on the natural ones. So Yeah, so what we did was to try to develop a computational method to actually build these immunoglobulin domains, controlling the overall geometry of the scaffold. So the idea, I will not go into details, but the idea was to generate new structures in which these loop conformations, those that connect the secondary structural elements, can be tuned to actually to tailor the overall geometry of the scaffold. And in this way, what we can do is to generate libraries of new immunoglobulin domains. And here it's a representation of the conformational space that is represented by natural immunoglobulin domains, and this is the space of computationally designed immunoglobulin domains. And in this way, you can see that you can explore a much larger conformational space, and also that is different to what you see in nature. So in this way, you have more control over the structure of the immunoglobulin domain, and potentially with the ability to incorporate different functionalities and properties. So for one case, for example, I can show you experimental results that we got for our best hit. This was a scaffold that was, well, with a good prediction. And then, as I said, no, so these proteins, we express them in E. coli and then purify them to actually verify their properties. So this is a protein, this is a circular dichroism spectra at different temperatures. And here, well, this indicates that the protein is well folded, adopts an all beta structure, and that is thermostable because the signal doesn't change with temperature. The protein is stable in solution, it's monodispersed, this is in chromatography. And then in collaboration with the lab in our same department, we managed to determine the crystallographic structure of this design. You can see the design in green, the crystal in gray, and the overlay of the backbone structure is very accurate. So it was close to 1 angstrom RMSD. So it indicates that that the experimental structure is validating our design model, and it was recapitulating many of the features that we had in the terms of the side chain interactions in the core and so on. So often what we want is always to have this experimental structure. Although we can do many very accurate predictions, we always need to perform the experimental characterization to actually corroborate the design model. So this was encouraging, and then towards this idea of developing antibody-like proteins, so we need to incorporate functions into that, right? So we need to incorporate, as we see in antibodies, those flexible regions that can perform binding events. So for example, what we did as a proof of concept, what we did was to incorporate a calcium binding motif. It's a flexible loop that has a sequence motif that can bind metals, and specifically calcium and terbium, and then we can perform binding assays in which allow us to actually measure this binding event. And we could see actually that the protein was well folded in the presence of this binding region and was active. And then after this, we decided to take like a second step. And then, as I said before, so in nature we have these variable domains that are these immunoglobulin dimers that pack face to face. This is what we see in this here, and this is the domain that sustains the flexible regions and has this arrangement in nature. This is consistent. This is conserved across nature. And then we reason, why not trying to design a new variable domain, but instead of packing what is known as face to face, doing edge to edge. So in this way, what you can do is to have hydrogen bond pairing between the beta strands and have a larger and continuous beta strand. This could be an interesting property because in this way you can minimize, for example, aggregation propensity. This is a problem we often have with this type of beta sheet proteins because these exposed beta strand edges to the solvent, this is what tends to oligomerize. And this, for example, has a high propensity to form amyloids when you have increased protein concentrations. So when you're trying to protect these edges and try to expand the hydrophobic core, making this type of arrangement, what is not seen in nature. So we developed a series of rules, and then what the idea was to design these arrangements, but in a way that you can fuse these two domains in a very structured way with very short connections. Because if you can do short connections, you can minimize the overall flexibility of the system. You can make it more rigid, and probably more stable. So these are, yeah. So so the idea was to generate different versions of this type of new arrangement. This is what we call as single chain immunoglobulin dimers, because these are two monomers that are connected in a single polypeptide chain. So yeah, so this was a study that we published this year. With two colleagues from the lab. So we generated these new type of designs. Here I wanted to show you that with AlphaFold we can make very accurate predictions with very high confidence of all these models. Again, we went to the wet lab to test the top predicted hits, and then we could see that we can also perform good predictions. And then by circular dichroism we see hyperstability with temperature, and also with chemical denaturants, which are a stronger. Well, it's a strategy also to trigger unfolding of proteins, also with chemical denaturation, and it's also a complementary assessment of hyperstability. And then for one of these cases, we managed to determine the crystal structure, and again, we compared the model with the X-ray. There was an excellent agreement in both structures. So this was a very good result. And then from there, following from the previous study, So now here I want to show you that, as I said, so we are developing this new type of arrangement for the variable domains. And then what we did was to try to see if we could actually find this type of arrangement in nature. Now, as I said, so we have the PDB, the Protein Data Bank, and the AlphaFold structure database, in which we have millions of predictions for many sequences. So now we reason it would be more likely to try to identify something in the AlphaFold database that has so many sequences. But it turns out that we cannot find anything similar either in the PDB nor in the AlphaFold database. We can find partial similarities in some of the domains, but the overall arrangement and having this continuous hydrogen bonding pairing and so on, it's very hard to see in nature. So in a way, we managed to create something that hasn't ever seen in nature and has very, very good properties. So yeah, so this is the final result. Here the idea is, okay, if we have this two-domain protein, why not trying to incorporate multiple loops? So then when we did a test to incorporate multiple functional loops into different areas, because the protein structure is very stable, it should sustain them. And yeah, we could see that we have been testing up to 2 loops, and we can see that they both are functional and the protein remains stable. So this is really encouraging. So now we are developing methods to actually develop designing these loops to actually be in protein targets, and then try to do the things that are even more similar to antibodies. And this is just a final slide just to show how we can diversify also these new type of scaffolds with deep learning, with hallucination strategies I mentioned before, in which we can develop more complex interfaces between the domains without any pre-specification, and we can generate hundreds of new proteins with very accurate predictions. And then with different shapes that can allow to tune the functional sites. So just to finish, I try to convey the message that we can now make very accurate predictions of protein structure at very low computational cost. So now anyone can do that. And in the same way, we have leveraged all this progress in this field to actually now design entirely new proteins that are not seen in nature, and that open so many new opportunities for biotechnology and medicine. But still, I would like to emphasize the importance of combining computation and experiment, because although these methods are being more robust, there is always the need to actually do experimental testing, to actually validate them and actually improve these models, right? And also because there are so many challenges still, for example, to infer, for example, protein dynamics from the structures, because many proteins, for example, in nature, not only allow one state, they need to exchange between multiple states. This is something that currently it's very hard to predict, or the effect of mutations, for example. This is another field that is requiring a lot of progress. So still there are many challenges ahead, but I wanted to say that it's a very exciting time to work on this. Yeah, and just to finish, I always like to show this sentence by Richard Feynman that had won the Nobel Prize. Because protein design and protein structure prediction are two complementary problems. And if we manage to design proteins, it means that we are starting to understand something about what it means to, what are the underlying bases of protein structure and how proteins fall. So in a way, protein design it's not only for applications, but also allow us to deepen our understanding of how sequence and structure are interrelated. And just I would like to finish thanking people from my team at IBMB, PhD students and postdocs, collaborators from the institute that allow us to complement our experimental characterization of the designs, and also external collaborators from the past and present, and well, funding agencies, and to all of you for your attention.
Okay, thank you very much for this very interesting talk. So now it's time for questions. Okay, well, many questions today, so it was a good talk.
What motivated you to come here and explain this interesting presentation to us? And what is the final message or idea or concept do you wish we remember the most today, apart from the ones you just mentioned?
Can you repeat, please?
What motivated you to come here and explain this interesting presentation to us today? And second part, what message or final idea do you want us to remember today?
Okay. Well, what I try, as I said, it's always very exciting to come to university and to talk to new students that are starting their career. And here I was excited to share my passion for this research field. I've been working for this for 14 years already, at least for the protein design part. And really I wanted to convey the message that now we are living like a privileged moment in science. So there has been so many ongoing developments in just very few years that now it's really exploded, the number of possibilities that it's really open. So I encourage you to further explore these ideas, whatever you read, because you are going to find motivations and many interesting challenges probably for you for the future. Yeah.
So what do you think are the main traits someone should have if they want to get involved or work in this field, especially when we are talking about research, innovation, and developing and designing from scratch?
I need you to reformulate the question, please. I don't hear you well.
When we talk about this whole field, like research, innovation, developing, and designing from scratch, what do you think are the main traits someone should have if they want to get involved?
The main traits? The main traits to get involved. What do you need?
Someone should have if they want to work in this field.
What you need if you want to work in this field? Okay. Well, as I showed, it's a very interdisciplinary field. We use computational methods for performing molecular simulations. This is one thing. Others for programming, so you need to program your pipelines to actually set up your calculations, to analyze the results. I would say you also need to have probably a more open mind in the sense that you need to understand a little bit of the experiments, because as I said, you want this interplay between computation and experiment. So it's always good to be always interested in the experiments, how they are performed, even if you have the chance to actually perform some of the experiments. In my case, during my I did a PhD in computational chemistry. I didn't have an experimental background. I went to do my postdoc in the Baker Lab, and there people were doing both. So you would be doing your models, computational design, and then you would do the experiments of you test your own proteins. So I had to learn actually how to experimentally do the proteins. It was challenging, but it was very rewarding, because then you validate the proteins that you have been designing in the computer. So I would say you have to be willing to learn so many things, and also to adapt to a very changing field. So in just a couple of years, we had to reformulate I would say 90% of the way we were doing before this advent of deep learning. We had been doing the things in the same way for, I would say, 5-6 years, but now with these methods you have to reformulate, and this is going to change even more rapidly. So we need to adapt very quickly and be aware of all the innovations that are in the field.
It is possible not to find the unknown structure despite having all this information?
Not having if it's possible not find an unknown...
Yeah, the unknown structure.
Well, it still is possible to find many novel sequences, many structures. As I said, no, sequence space is huge. So yeah, and even if you want to perform even more complex functions, I would say totally, yeah.
If degenerative diseases such as Alzheimer are caused because of the accumulation of protein between neurotransmitters, how could a protein be created or modified with the Novo design of proteins to avoid this accumulation?
Okay. So so there is people working on that. So as other diseases, right? So for example, you want to block a protein-protein interaction in this case. No? In this case, you want to block the amyloid formation, that is interaction between between beta strands in that case. So there is people actually developing, for example, proteins that can mimic. Often what you want to do is to mimic that interaction in a different scaffold so that you can compete with the natural partner. So so this is something that is being addressed. Another thing, for example, for Alzheimer's disease in particular, it's you have other extra challenges that is crossing the blood-brain barrier. Still, many of the things still need development on actually how to incorporate proteins inside the cell. This is or across tissues. This is something quite challenging. But yeah, certainly the field is going towards that. Yeah.
Do you think new design proteins could replace natural proteins, for example, in the human body in the near future?
Well, for example, so traditionally there has been, for example, what is known as the replacement therapy. No? So you have a natural protein, or you lack, for example, a natural enzyme, an enzyme that you should have in your body, but you had a genetic disease, and then this enzyme is not present or is non-functional. So for ages, no. So there has been this approach to actually to replace the enzymes. But for example, there can be a problem that this enzyme is hard to produce or it's very unstable. So this could be a way to actually to develop proteins that have similar property but with improved functions. One of the things also that you have to take into account is also issues of immunogenicity. That's something that every time needs to be validated. So every time you design a new sequences, although there are very promising studies in vivo that show that they don't show remarkable immunogenicity, but this should be something that should be checked before, right? But in principle, yeah.
No? Okay, if not, let's thank again Enrique Marcos.